Hello and welcome to the pharmaphorum podcast. I'm your host, Jonah Comstock. Genetic medicine has tremendous potential, but it is also expensive to develop for each condition. And there are a huge number of conditions, including rare diseases that affect very small numbers of patients. So my guest today is Michelle Werner, CEO of Alltrna, and she's going to talk to us about how her company is creating a new approach to genetic medicine that doesn't go one disease at a time. And the key to that is transfer RNA, hence Alltrna, all tRNA. So Michelle and I met recently at a flagship pioneering reception, and I was very interested in what she had to say, and I'm excited for her to share it with all of you. So welcome to the show, Michelle.
Thanks so much for having me, Joan. I'm looking forward to this conversation.
So maybe just start with the elevator pitch a little bit. Why does tRNA enable you to kind of just completely shift the paradigm on genetic medicine development?
Yeah. So you mentioned a little bit about this in your intro, right? As you know, there are somewhere around 10,000 different genetic diseases, which is a lot, especially when you consider the fact that modern technology that we have access to today, whether it's mRNA, gene therapy, gene editing, siRNA, kind of fill in the blank, all must take a disease by disease approach because that's how those technologies work. They're specific for a certain gene or a certain protein, or even in the case of gene editing, a specific mutation within a specific gene. So we've been trying to tackle the problem of 10,000 or so different diseases one at a time, which you can imagine from a patient perspective is a real dilemma. Because if you're one of those patients or a caregiver of a patient who has one of those diseases that maybe are less common, not amongst the top 10, 20, 30 of those different diseases that are the most common, the likelihood of having an individual therapy for those diseases in that person's lifetime is infinitesimally small. And so for us, that's where we believe the tRNA has this unique potential, because one engineered tRNA may have the opportunity to address hundreds, if not more, different diseases. Because a tRNA, in the way that it functions, which I'll come back to in a second, works independently, the same way, independently of the gene, independently of the protein that's being coded for, and independently of, kind of location for where mutations that are the genesis of different conditions occur. So that location is also irrelevant. So the tRNA, let's think about the basic biology and its function, because not everybody recalls what a tRNA actually does. So in the protein translation process, the tRNA plays this hugely important role where it deciphers the code for each and every amino acid that's being coded for in the mRNA strand. It finds that amino acid in the surrounding cellular environment and transfers it to the growing polypeptide chain. And what's really important to note is that tRNAs do this job, as I mentioned, independent of gene, independent of protein, independent of, the different codes along the coding sequence and mutations that occur in the coding sequence. Now, what a tRNA or an engineered tRNA has the ability to do is actually address the mutations that are common across all of these different diseases. So instead of addressing one disease at a time, we could address the mutations that are the same actually across thousands of different diseases. We observe the same mutations in upwards of 4,000 or so of these different genetic conditions. And by sort of taking the gene and pushing it to the side and focusing on a mutation-specific approach, we have this ability to group together patients that have those common mutations independent of the disease or independent of the gene that's affected. And that is what allows it to play this very unique universal role in addressing those patients' conditions. That is not the case for any of those other genetic modalities, gene therapy, gene editing, mRNA, for example, which really sets a tRNA apart.
So I think we discussed this when we first met. The thing that immediately comes to mind for me is that it feels like our systems of regulation are not designed for something like this. Even if this can address a lot of different conditions, you still have to, in order to make it prescribable for all those conditions, you have to test it in all those patient populations. I mean, is that the case, or is there maybe a creative pathway I'm not thinking of?
Yeah, so traditional drug development would lead one to believe that that is the case, Jonah. So, I mean, with all those other modalities and all, many small molecules, it's the same way as well. It's a really one drug, one disease technology, and therefore the regulatory systems are also one drug, one disease. Now, if you look at certain examples in, let's say, oncology, we see places where that is not actually the case. And for example, there are a growing number of what I would call platform trials that could be either a basket trial design or an umbrella trial design. And if we just think about what a basket trial is for a second, and that's relevant for our development pathway, that's where you have patients who technically have different types of disease, but they're all given the same treatment intervention, and patients are measured using, similar outcomes, or you put it into your plan about how you would analyze the patient's outcomes accordingly. Now, this is used over and over again in the oncology space. In fact, there are, dozens of different drugs that have been approved using a basket trial setting. Most of them are in the oncology space, but we actually do see some examples for small molecules in the rare disease setting as well. Now, a tRNA technology could very readily leverage this type of development strategy and therefore the regulatory strategy accordingly in order to be able to have this be a path forward for us. And we think, honestly, we haven't seen as much of this in the rare disease space because, quite frankly, as I mentioned, no technology or very, very few technologies have the ability to do this. So there hasn't really been a need to I guess, really formalize that as a strategy, both from a development perspective as well as a regulatory perspective in the rare disease setting until perhaps now. And the tRNA may be the, I'd say, inspiration for that to become more formalized. Now, the methodology is already there, and as I mentioned, has already led to approved medicines using a basket trial strategy. So we're not completely reinventing the wheel and starting from scratch. We're just applying that approach in a way that hasn't been possible before.
So, with something novel and innovative like this, I always ask, why hasn't anyone done this before? What have the challenges been, or was there something that you had to sort of see that no one had seen before?
Yeah. So maybe if I sort of go back a few years to the founding of the company, which actually took place in 2018, it starts with a process that, flagship pioneering is known for by asking a big what if question that perhaps is not a question that has been asked before in the past. And the what if question was, well, what if you could leverage a tRNA to alter the protein translation process, right? And really, I'd say sort of highlight its role and its importance more so than perhaps has ever been done before. And that was the first thing that we asked. And actually, what we quickly discovered, and not myself because it was before my time, but the founding scientific team at Flagship Pioneering really discovered that the fundamental tools to quantify tRNAs, to characterize tRNAs, to differentiate an engineered tRNA from an endogenous tRNA, they didn't exist. And so because the tools didn't exist, maybe that also was a barrier in terms of people asking that question and trying to figure out whether or not there was a there, let's say, with the tRNA. And so the founding scientific team actually had to start with putting some of these tools together. And that's exactly what we've done, put together a suite of proprietary tools. I think that Alltrna might have been the first company to actually chemically synthesize an oligonucleotide version of a tRNA that isn't just a tRNA, but also has chemical modifications. And I can come back to that in a second, to really design a tRNA in a way that we think could have therapeutic potential. So we may have been the first to be able to do that. And then we had to figure out the tools that we needed to, as I mentioned, quantify them and, characterize them and be able to differentiate them from endogenous tRNAs. And once we had that suite of tools together and knew that we could actually chemically synthesize these things, that really became like, oh, wow, right? Like maybe there's potential here. And that really was the kind of early days that was the beginning of us on the journey where we are today, where now I actually think we're very close to demonstrating that this has the potential to be a therapeutic for patients.
This has a sort of serves as a good case study for the flagship model, right? Because you're in a situation where in order to build the thing that's going to have a therapeutic implication, you have to build the tools, you have to almost invent a whole science. And there's, I'm sure, so much capital required to that before you are anywhere near seeing a return, even more so than normal biotech. But I know Flagship has sort of ways to sort of spread around the capital that allows them to make big investments like that, right?
I mean, definitely being pioneers comes with a cost because it takes the right mindset to, ask those bold questions. It takes the right, knowledge base to be able to say, okay, how do we tackle these challenges and really do something that maybe nobody has ever done before? And yes, of course, there's capital requirements that are needed in order to be able to support those efforts. And I do think that we're very fortunate that Alltrna is a flagship pioneering company, has been able to access, both knowledge and expertise as well as financial backing to really demonstrate the potential of this technology.
Talk to me about sort of what else is required to get from here and there, how far along you are um,
Yeah.
in something that can get into clinic.
Yes. So we're getting close, Jonah, which is really exciting. So I will say that over the course of the last, so as I described the sort of founding scientific team's work, really putting the tools together, making sure that we can synthesize these things, the next couple of years really was trying to prove the preclinical proof of concept. And we've done that now. So what I mean by that is, and I'll just share with you, the first application that we're thinking of for a tRNA is reading through nonsense mutations to restore full-length protein production. And a nonsense mutation, I'm sure as you know, is when one of these codes for an amino acid codes for a stop instead. And so instead of having a full-length protein, you have a protein that's too short or often actually absent altogether, and that's what causes disease. We would actually classify any patient as having one of these stop codon mutations as having stop codon disease. And that roughly accounts for about 10% of all genetic diagnoses, which could be somewhere around 30 or 35 million people worldwide. So a pretty significant patient population if you think about it. So what we then did over the sort of next couple of years is demonstrate from a preclinical perspective that we could use the same engineered tRNAs to read through these stop mutations across a number of different diseases. And in a cell model, we've looked at this now in over 25 different cell models, different genes, different proteins that are being coded for, and actually different locations for where mutations occur in a coding sequence, all using the same engineered tRNA. And we have been able to demonstrate in those models the read through of the premature termination codon and the restoration of the full length protein. Now, that's a cell model. Now, next step would be to demonstrate exactly that in an animal model. And we've now done that in 3 different animal models. We've actually disclosed data from 2 different animal models. One is a methylmalonic acidemia animal model, and the other is phenylketonuria. Both of these are rare genetic liver diseases, and that's an important point to make because our first engineered tRNA is formulated in a lipid nanoparticle, and therefore lipid nanoparticles being very effective at getting to the liver, we're really targeting liver diseases in that first instance. And so we've started to look at demonstrating preclinical POC in those disease setting. And what we've demonstrated with AP003, which is our lead development candidate that's advancing towards the clinic, that those animal models both have the same nonsense mutation, and AP003 reads through those nonsense mutations in both MMA and in PKU and in another disease that we haven't disclosed yet, um, and restoring full length protein and therefore addressing the underlying nature of the disease. Now, again, I don't think I think we were the first to do that. And we were first to do it across multiple disease models. And we're first, to do that with, a single tRNA. But it really speaks to the concept of instead of single drug, single disease, single drug, many diseases, in fact.
Yeah.
And so that's really what we've done over the course of the last couple of years. And once we realized that AP003 has the potential, we believe, to be clinically relevant or therapeutically relevant, we actually started the IND enabling work. And we're in the latest stages of the IND enabling process right now as we speak. And I'm really excited to say that I believe that 2026, this year, we should be entering into the clinic and should be able to dose our first humans with a tRNA, which will be the first time ever.
Wow. So, I mean, in a lot of drug development, there's a little bit of skepticism, that models will translate to human models. In genetic medicine, it does kind of seem like, a gene is a gene. It, I mean, I would think limited lame person's knowledge that there'd be very strong reason to believe that if it works in animals, it works in people.
I mean, if you look at RNA medicines in general, that has historically played out, right? So I think a lot of that is because the biology behind these diseases are very highly conserved and they translate, they're similar in different species. And actually, the biology behind a tRNA is also very highly conserved and is very much the same from species to species. So if you think about conserved biology of both the disease as well as the modality, one would believe that there's a high likelihood for that to translate clinically. But of course, until you run the experiment, we don't know. And that's really the importance of doing human studies, is to really validate is what we're seeing both from a safety perspective, but as well from an efficacy perspective in animal models, similar in humans. And that's what we look forward to being able to do in the clinic.
Let's talk a little bit about the patients that this is going to affect, because this is really an option where there's just no options, right? And you were saying there might never be any options if you have an extremely rare condition. I did a podcast probably 2 years ago that still really stuck with me with some parents of a child who had a genetic disease, and they were crowdfunding development of a treatment for her specific disease because that was the only way. So, I mean, it really is, these patients are in a tough spot right now. And I think it's just worth sort of talking a little bit about, who you're hoping to be able to help here.
I mean, so this, I'm so glad that you brought this up, Jonah, because it's the patients that is really what drove me to Alltrna in the first place. You just referenced these parents that you spoke to in a previous podcast who, I mean, I've met so many of these families myself as well. You may know this, Jonah, about me. I'm not sure, but I also am a rare disease parent too. I have a son with Duchenne muscular dystrophy. And when he was diagnosed 5.5 years ago, um, I was gobsmacked really about the limited options available for him. I mean, the standard of care at that time had not changed in over a decade. And he was not eligible for any clinical trials, even though there is more sort of innovation happening in the Duchenne space than there is, and maybe I'd say the vast majority of rare diseases. But still, regardless, he wasn't eligible for any clinical trials. So I know what it feels like to feel like you're overlooked, your options are extremely limited. And unless you try to figure out a path forward yourself, that path is not going to magically appear. That's how it felt when my son was diagnosed, and I'm sure that's how it felt for these families that you spoke to. I have met so many families who have children or a loved one who have one of these ultra rare diagnoses. And unlike myself, who went to clinicaltrials.gov and did find some Duchenne trials, many of those families will find absolutely zero clinical trials. Um, and honestly, that is because the traditional drug development pathway really only allows for drug development in the most common of the rarest diseases. And I think that is so tragic because that means that so many of those patients are just going to continue to be overlooked. And actually, it's one of the main reasons why only 5% of these genetic diseases have an approved drug, because we're only focusing on the ones that are most common. That means 95% of those diseases, have nothing and will likely still have nothing unless we figure out a different way to tackle them. And that's really what I got so excited about the tRNA and Alltrna's approach, specifically when I was first introduced to the company and my own family's experience, but also having connections with many other rare disease families as well. It really made me realize that this could profoundly benefit a population that would likely continue to be underserved if nobody does it any other way.
Well, it's exciting to be doing work that has the potential to make such a big difference for people.
It is.
What haven't we talked about yet that you think is an important part of Alltrna, your story, your mission, whether it's more on the scientific side or more on the around some of these other aspects?
Yeah, I mean, I guess one thing that I would maybe also just highlight is not all tRNAs are created equal. And there's a lot of work that goes into the design of these tRNAs that I think makes a big difference in terms of their clinical potential. And maybe just to put things quite simply, there are sort of three ways in which tRNAs can be designed, one of which is just engineering the anticodon to read the specific premature termination codon, the other of which is optimizing the sequence of the roughly 76 nucleotides that make up the tRNA structure. And then, as I mentioned earlier, these chemical modifications that really do enhance the tRNA's effects, such as their activity or their potency or perhaps even their stability. At Alltrna, we're doing all three. And I think we're the only group that is actually leveraging all three of those design principles. And what we've learned is all three of them play a very important role. And so we're really excited to see then how that plays out once we get into the clinic. That design matters just as much as the way in which we run a development study and bring this novel therapy to patients.
Awesome. Well, thank you so much. Look forward to hearing what happens in 2026 and getting to human trials. I know you mentioned before we started recording that you're going to be at JP Morgan next week. Probably by the time this airs, we will have already been through that. You have any news you're announcing at the show, or you're just there to kind of connect with people, learn what's going on related?
We're connecting and we're sharing our progress towards advancing to the clinic, just as we talked about today, Jonah. So really excited to be getting that message out there.
Awesome. Well, thanks so much for chatting with me today. Really, I was excited and interested in what you're working on when I met you at that reception event, and I'm very glad to have the opportunity to learn a little more. And good luck. It seems like a really promising space.
Thanks so much, Jonah. I appreciate it. We'll look forward to keeping in touch.
That concludes another episode of the pharmaphorum podcast. You can find more information about this episode, including a download link and information about previous installments, at pharmaphorum.com/podcast. The pharmaphorum podcast is also available on Apple Podcasts, Spotify, Overcast, Pocket Casts, Podbean, and pretty much wherever else you download your podcasts. Just search for pharmaphorum and follow and hit subscribe. Of course, don't forget to visit the website itself, where you can sign up for daily and weekly news and analysis bulletins, and follow us on Blue Sky and LinkedIn at pharmaphorum. That's PharmaPhorum. That's all for now. Thanks for listening.